This conference will now be recorded.
Today we are going to learn about 5 spotters. Okay, so it is practical exercise for you. We are going to learn 5 spotters. You have to write every point of the spotter in your record, as well as draw the diagram. These spotters will also come for you for examination. Okay, so let's now start the class. These are the 5 spotters that we will be learning today. Sterile swab, post nasal swab, while loop, Pasteur pipette, and Seitz filter. Okay, so these 5 spotters we are going to look at the picture properly. Did you see? It's a swab. Okay, so it is a sterile swab. So how it is this? So how will you identify? It is a wooden stick. Okay, on the stick, cotton swab is there. So this is a cotton swab. Points. Sterile swab. It is a cotton swab rolled on the stick and sterilized by hot air oven at 160 degrees centigrade for how many? 60 minutes or 1 hour. So sterile swab. What is it? It is a cotton swab which is rolled on a stick. Sterilization. How will you sterilize it? By keeping it in hot air oven and heating at 160 degrees centigrade for 1 hour. So, what type of a heating method is this? It is a dry heat sterilization method. Types of swabs. How many types of swabs are there? There are different types of swabs. First one, post nasal swab, perinasal swab, laryngeal swab, cervical swab, and high vaginal swab. Okay, so these are the common types of swabs which are present. Precautions. What are the precautions that you take? Unplugged swab tubes should never be used. What are swabs used for? To collect samples. Okay, as well as they are used for doing lawn culture also. Whatever it is, you must never use unplugged swab tubes. Why? If they are unplugged, then the swabs may have been contaminated. That is why compulsory while sterilizing, all the swabs have to be kept in a test tube and plug the test tube with a cotton plug and should be sterilized. Uses of cotton swab. It is used for collecting body fluids, pus, throat, cervical discharge, ear discharge, and other fluids from local lesions. Second one, for preparing lawn culture of bacterial growth. in the case of antibiotic sensitivity testing. Okay, so this is sterile cotton swab. Once again, how will you identify this? It is a stick on which a cotton is rolled. If it is that, then it is a sterile cotton swab. Write in it 2 points. First point, how will you sterilize it? You will sterilize it by heating in hot air oven at 160 degrees centigrade for 1 hour. Uses for collecting samples and also for lawn culture. Second spotter. See these two properly. Differentiate between cotton swab and this swab, how it is. Okay, the swab is present on a wire. Okay, so second one is post-nasal swab. What is it? Post-nasal swab, it is a 20 mm swab of a flexible metal. Terminal 20 mm of the flexible metal swab is bent at an angle of 45 degrees so that when introduced through the nose, it carries the swab up behind the soft palate into the nasopharynx. Okay, so post-nasal swab means it is a flexible metal swab. Then how is sterile cotton swab? Sterile cotton swab is present on a wooden stick and it is straight. Whereas post-nasal swab, it consists of a flexible metal swab and the end of the swab is bent at an angle of 45 degrees so that you can easily insert it through the nose. Precautions to be taken. Salivary contamination has to be avoided. Yes, while using post-nasal swab, while collecting the specimen, you have to avoid contamination with saliva. Next, the swab is contained in a stopper test tube of sufficient width to admit the bent end. Okay, not ordinary tube, but a specialized tubes are there in order to insert the swab so that the bent end has sufficient space. Next. What are the uses of post-nasal swab? It is used for the collection of sample material from post-nasal region, that is, nasopharynx, to detect the presence of nasal carriage of meningococci. For collecting specimens in case of pertussis, that is whooping cough. Not only this, post-nasal swab now from the nasopharynx, what do you use? You are using it to collect samples for COVID, right? From this region. So the swabs can also be used to collect viral specimens from nasopharynx for the detection of viral infections. So this is post-nasal swab. Okay, so differentiate between cotton swab and post-nasal swab. In the case of cotton swab, it is present on a wooden stick. A swab is present on the wooden stick and the end is straight. Whereas post-nasal swab, it consists of a flexible metal wire swab and it is usually bent at 45 degrees so that you can easily insert it into the nose. Next, spotter. Have you seen this? It's a wide loop. Okay, it's a nichrome wide loop. Nichrome wide loop or wide loop. Used to inoculate culture media, and wide loop may be of nichrome or platinum. So what is this wide loop used for? It is used to inoculate culture media. Material, it is made up of nichrome or platinum. The wire used is SW324, which is about 2.5 inches in size and held in a metal handle. The wire is sterilized by flaming. You keep the loop under the flame of the Bunsen burner and heat it until the wire becomes red hot. That is how you will sterilize wire loops. Uses. What are the uses of wire loop? In the case of platinum wire loop is used for special tests such as oxidase test and catalase test. Okay, oxidase and catalase tests are biochemical tests used for the identification of bacteria. Then we use, and when we want to detect bacteria by performing these tests, we have to use platinum wire loop, whereas nichrome wire loop is used routinely. Means what? For all type of streaking cultures and everything, you will be using commonly nichrome wire loop. Only when performing oxidase and catalase test. Instead of using nichrome wire loop, you use platinum wire loop. Other types of wire loops are straight wire. Straight wire is used for stab culture. Means what? Agar media is usually present in culture tubes. Okay? You pick up a colony and just stab it. Means you just pierce it. So for stab culture, straight wire is used. Next, L-shaped wire. L-shaped wire is used for fungal inoculation. When we want to culture fungal specimens, then we usually use L-shaped. You know how is a loop? A loop is usually round and the end is round. Whereas straight wire means no loop, just straight it will be. L-shaped means it will be bent. Okay, so what is this wire loop? Okay, what is this wire loop? It is used to inoculate culture media, and there are two types of wire loops. They are platinum and nichrome wire. The sterilization is by flame. Next, what is this? It is a Pasteur pipette. Okay, so this is a Pasteur pipette. What is it? It is an ordinary glass tube where one end is elongated into narrow thin part. On the other end, a rubber stopper is there. So this is Pasteur pipette. Pasteur pipette. It is the simple glass tube drawn into a long tapering end. Yes, we have seen the diagram now. It is an ordinary glass tube where it is drawn into a long tapering end. Uses: it is used to collect body fluids and pus from deep areas for bacteriological investigations. Commonly, Pasteur pipette is used to collect body fluids and pus from deeper areas. If you can't collect them by swabs, then you use Pasteur's pipette to collect. Next, it is also used to deliver solutions or reagents in test tube containers containing various processors, and also for the inoculation of pathological fluids like urine, cerebrospinal fluid, and exudate. We sterilize Pasteur pipette by hot air oven at 160 degrees centigrade for 1 hour. Okay, so Pasteur pipettes are sterilized in hot air oven. Fifth spotter. So what is this? It is Seitz filter. It is used for filtration. Seitz filter. It is a filter made up of asbestos used for filtration of liquid media. So what is this? It is a filter which is made up of asbestos, which is used for liquid media. The Seitz filter consists of a metal container with a holder to keep the asbestos filter. The liquid to be filtered is passed by either positive or negative pressure. Okay, so this Seitz filter consists of a metal container. Inside the metal container, you usually keep the asbestos. What is it used for? It is used to sterilize fluids that cannot be sterilized by heat. Okay, so this is mainly used for the sterilization of fluids where you cannot sterilize by heat. Example, serum, toxins, antibiotic solutions. All of these are used to sterilize by using Seitz filter. However, there are certain advantages of using Seitz filter. What are they? First one, high absorbing capacity of asbestos tend to alkalinize liquid filters. Means what? The asbestos filter has a very high absorbing capacity. As a result, it will usually alkalinize the liquids which are filtered. And also, it has carcinogenic potential. So this asbestos filter is also carcinogenic, therefore not used at present. Okay, so what is a Seitz filter? It is a filter made up of asbestos which is used for the filtration of liquid media. Okay, how is the apparatus? It is a metal container, and inside the metal container you keep the asbestos filters. What type of solutions are used by filtration by asbestos? Substances such as serum, toxins, and antibiotic solutions are all used by filtration method. What is the disadvantage of using Seitz filter? Seitz filter usually uses asbestos, right? So asbestos has a high absorbing capacity. So when you filter a liquid, that liquid may become alkaline. And also, asbestos has carcinogenic properties, means cancer-causing properties. Okay, so these are the two disadvantages. Okay, students. So these are the 5 spot tests for today: sterile swab, post-nasal swab, wine glue, Pasteur pipette, and Seitz filter. All the 5 you have to write in your record in spotters page as well as draw neat diagrams. Okay, that's all for the class today. Thank you.